Welcome to the deep dive. Today we are taking a massive shortcut right into the blueprint of life, the entire kingdom Animalia.
Yeah, the whole thing.
We're zooming in on the defining characteristics, the grand evolutionary timeline, and, the basic architectural plans that allow for this staggering diversity of animals, from, like, the smallest nematode to the biggest blue whale.
This is a really foundational deep dive. Our mission, I think, is to distill the core concepts of animal biology. We want to structure the information so you can quickly and thoroughly understand what truly separates the animal kingdom from everything else.
And how that separation led to the most dynamic life forms on Earth.
Exactly.
So let's start with the most basic definition, the one thing we all share. Animals are the ultimate consumers. We're the kingdom of heterotrophs.
That's the critical functional distinction right there. While plants make their own food and fungi, they absorb nutrients externally, we operate differently.
Totally differently.
Animals get energy by eating other organisms, by ingesting that food, and, crucially, processing it internally, usually with a really efficient digestive tract.
You see that everywhere, that instinctual consumption in, like, the rapid strike of a chameleon's sticky tongue grabbing a moth. But the efficiency is also internal, right? We have this tube, the digestive tract, with a mouth at one end and an anus at the other. It's just designed for this continuous, specialized food processing.
Absolutely. But, consumption alone doesn't define us. What truly sets the animal kingdom apart is the structural and developmental fingerprint.
Okay, so let's unpack that. The structural profile. We know animals are multicellular, heterotrophic eukaryotes, but what's the cellular glue that holds us together?
So the key structural difference is that animals lack the rigid cell walls that you'd find in plants and fungi.
Right.
Instead, our cells are supported and connected by these complex structural proteins outside the cell membrane, and the most famous one is collagen. Yeah, think of it as the essential scaffolding that allows animal cells to be flexible, to move, and to adhere to form complex tissues and organs.
And that flexibility, that must be what enables the two specialized tissues that are so central to the animal lifestyle.
The unique nerve cells and muscle cells, yes.
Nerve cells for communication, muscle cells for movement. This combination is what fuels, every observable animal behavior, from flying to hunting to just navigating.
That's why movement is so synonymous with the animal kingdom. And where these sophisticated tissues come from is rooted in a really specific, repeatable pattern of embryonic development. That story is our most reliable signature.
All right, so walk us through that initial architectural sequence. Let's start with the fertilized egg, the diploid zygote.
Okay, so the zygote first undergoes what's called cleavage. Imagine rapid, successive mitotic divisions. But here's the crucial twist.
What's that?
There's no cell growth between the divisions. It's like the embryo is just shrink-wrapping its contents, resulting in smaller and smaller cells packed into the same volume.
Okay, so just dividing, not growing.
Right.
And this rapid division leads pretty quickly to the blastula stage, which, can we visualize that as like a hollow soccer ball?
That's a great way to think of it. It's a sphere of cells surrounding an internal, fluid-filled space called the blastico.
And then comes the truly formative event.
Gastrulation. This is where that hollow ball of cells folds inward, like you said, like punching an indentation into that soccer ball. And this folding produces the embryonic tissue layers, the germ layers.
So that indentation is creating the foundational blueprints. The outer layer is the ectoderm.
Right, which will ultimately become the animal's outer covering, its skin, and critically, the nervous system.
And the innermost layer, the one lining that new internal pouch, the archenteron, that's the ender.
Exactly. And that forms the lining of the digestive tract and associated organs like the liver or lungs. Everything else is kind of built around those two layers.
It's also worth noting that a lot of animals don't just, grow up. They change dramatically. They often start as a distinct larva.
Sexually immature, eating different food, living in a different habitat.
Right, before undergoing metamorphosis into a juvenile or adult form.
And this entire complex sequence, across species as different as a clam and a human, is regulated by this ancient genetic toolkit. We're talking about Hox genes.
Okay, so here's where it gets really interesting. These are unique homeobox-containing genes. They're master regulatory genes.
They control the expression of hundreds of other genes, basically dictating the development and the overall body plan.
The wild thing is how conserved they are.
It's incredible. The same family of Hox genes that sets up the head-to-tail axis in a fly is operating, with modifications, in a mouse. This vast complexity of form is governed by a set of genes inherited from an ancestor hundreds of millions of years ago.
And that idea that we're all working with variations on the same ancient instruction manual, that brings us to the history of animal life. Once we get what an animal is, the natural question is, when did it first show up?
Well, the deep timeline is staggering. Molecular clock estimates, which analyze rates of DNA change, they place the common ancestor of all living animals at about 770 million years ago.
And we have physical proof for that, right? What's fascinating is the first biochemical evidence of animals. It's not a hard fossil, but a molecular signature.
Right, researchers found these unique fossil steroids in 710 million-year-old sediments. And these steroids are primarily produced by a specific group of sponges.
So it's chemical proof that animals were already swimming around way back then.
It is. And this leads us backward to the single-celled origin. The closest living protist animals relatives to animals are the choanoflagellates. And this isn't a guess. It's a molecular connection based on shared machinery.
For cell adherence and signaling, right?
Exactly. Choanoflagellates and animals share protein domains, like those used in cadherin proteins, which are essential for cells to stick together.
But here is the critical evolutionary nugget. Animal cadherins didn't invent a new protein from scratch.
No, they simply incorporated a novel domain, the CCD region, into the existing choanoflagellate protein structure.
So it's descent with modification through genetic tinkering.
That's it. Evolution wasn't always about inventing new tools. A lot of the time it was about rearranging the parts of ancestral tools they already had to achieve something new, like multicellularity.
Okay, let's move forward through the major geologic milestones. Starting in the Neoproterozoic era, spanning from a billion to about 541 million years ago.
This is where we see the macroscopic, soft-bodied organisms known as the Ediacaran biota emerge, around 560 million years ago.
But the real shift, the game changer, comes with a small fossil called Cludina.
Yeah, this animal had a shell, and some of the fossils show these small drilled boreholes. This tiny detail is huge.
Because it's early evidence of predation. Something was drilling through the shell to eat the animal inside.
Exactly. And that single fossil marks the start of this evolutionary arms race. Protection selects for attack, and attack selects for stronger protection.
And that arms race, it's possible it fueled the geological blockbuster that followed. The Cambrian explosion.
That event, early in the Paleozoic era, was an astonishing 10 million year period, from about 535 to 525 million years ago. We just see this rapid appearance of most of the animal phyla we know today.
Including the first large animals with hard skeletons, and most of these fossils are bilaterians.
Right, bilaterally symmetric animals with complete digestive systems.
So why the burst? There are a few leading hypotheses, and they're all kind of interwoven. First, as you mentioned, that escalating predator-prey coevolution. If you're a hard-shelled animal, you survive. If you're a predator with sharp claws, you thrive. It drives rapid adaptation.
Second, there was a major increase in atmospheric oxygen around this time. And why that matters is that higher oxygen levels can support animals with higher metabolic rates.
So if you want to be a big active predator,
You need efficient metabolism, and that requires more oxygen.
and finally, the genetic hypotheses. The origin of new Hox genes, or maybe key regulatory microRNAs or miRNAs, provided the necessary genetic flexibility to generate all these new, complex body forms so quickly.
Yeah, the Paleozoic just kept going. Arthropods were the animal pioneers colonizing land around 450 million years ago. So think ancient millipedes, early spiders.
And vertebrates, they took their time. They only arrived on land around 365 million years ago, then diversifying into tetrapods.
Then you hit the Mesozoic era, 252 to 66 million years ago. This is the age of the giant reptiles, the dinosaurs.
The origins of birds and the first mammals, which it's funny, there were these tiny nocturnal insect eaters just hiding in the shadows.
That era, of course, ended very abruptly with the mass extinction. That event cleared the playing field leading into the Cenozoic era 66 million years ago to today, where the loss of the large non-flying dinosaurs allowed for the rise of the large mammalian herbivores and predators that we see today.
It's a huge amount of history, but the result is this massive kingdom defined by structure. And this brings us to concept 3, understanding body plans, the architectural guidelines that animals follow.
A body plan is just that integrated set of morphological and developmental traits that define a lineage. And the easiest way to start categorizing them is by symmetry.
We see two main types. First is radial symmetry, like in jellyfish or sea anemones, where the body parts are arranged around a central axis. A jellyfish doesn't really have a front.
And that symmetry is perfect for a sessile or planktonic lifestyle. If you're floating or you're anchored, you need to be able to interact with your environment and capture food equally well from all directions.
Then you have bilateral symmetry, which is what we share with almost all active animals. Lobsters, fish, mammals, we have two axes, anterior, posterior, or head-tail, and dorsal-ventral, top-bottom.
And bilateral symmetry is intrinsically linked to active movement. It promotes cephalization, the concentration of sensory organs and the central nervous system at the anterior end, your head. If you're hunting or moving forward, you need your processing power and senses pointing where you're going.
Right. And complexity also increases with the tissue layers. Diploblastic animals like cnidarians only form the ectoderm and endoderm.
But all bilaterians are triploblastic. They add that third layer, the mesoderm, sandwiched in between. This is the structural powerhouse. The mesoderm forms the muscles and most of the complex internal organs.
And within those triploblastic animals, we see variations in the body cavity. The existence and nature of this cavity is crucial for function.
Oh, absolutely. The most advanced is the calum, or true body cavity, which is completely lined by mesoderm-derived tissue. This cavity offers significant mechanical advantages.
Okay, give us an example. Why do we care about a calum?
Yeah, it does a few critical things. It cushions internal organs, protects them from impact, and it acts as a hydrostatic skeleton in soft-bodied animals like earthworms, so they can exert force and move.
And allows internal organs to grow and move independently.
That's the most important part, yes.
Yeah.
It lets them move independently of the outer body wall.
That's the one I love. Imagine if every single time your heart beat, your skin rippled. The calum prevents that internal organ activity from warping your external body, so you can run while your heart and digestive system are doing their own thing.
Exactly. Now, not all internal cavities are true colums. Some animals, like nematodes, have a hemicole. This cavity is lined by tissue from both mesoderm and endoderm, and it's filled with hemolymph.
Which is a fluid for internal transport.
Right. It doesn't offer quite the same structural advantages. And finally, you have the ecolomates, like flatworms, which lack a body cavity entirely.
But the most fundamental distinction in bilaterally symmetrical animals is how they develop early on. The difference between protostome development and deuterostome development.
And this split forms the two largest branches of the animal family tree.
Okay, so this distinction is defined by three key patterns. First, cleavage. Protostomes undergo spiral and determinate cleavage.
And determinate means that the fate of those early embryonic cells is fixed very, very early on. If you separate the cells at the four-cell stage, none of them can form a complete animal on their own.
But deuterostomes, on the other hand, use radial and indeterminate cleavage.
Indeterminate, meaning each cell retains the capacity to develop into a complete embryo. This is the difference that actually makes identical twinning possible in deuterostomes like us. It shows a flexibility in development that protostomes just don't have.
So the second difference is how the coelom forms. In protostomes, the coelom forms when solid masses of mesoderm just split open to form the cavity.
Whereas in deuterostomes, it's a bit more elegant. The coelom forms as outpocketings that pinch off from the wall of the developing gut, the archenteron.
And the final difference is the simplest to remember, and it's right in the name. The fate of the blastopore.
That initial indentation during gastrulation.
In protostomes, which means first mouth, the blastopore becomes the mouth.
And in deuterostomes, meaning second mouth, the blastopore becomes the anus, and the mouth forms secondarily later in development. This is a critical distinction that dictates how we draw the whole evolutionary tree.
And that roadmap brings us to our last bit here, concept 4, the clades of animal phylogeny. This summarizes these vast relationships based on both structure and molecular data.
So all animals are in the clade metazoa. At the very bottom, forming the sister group to all others, are the sponges, or porifera. They're largely defined as the basal animals because they lack true tissues.
Okay, moving up from there, animals with true tissues are in the clade eumetazoa. This includes the basal eumetazoans like cnidaria, jellyfish, which are diploblastic and radially symmetrical.
But the massive, sprawling majority of eumetazoa is the clade bilateria, all the triploblastic bilateral creatures, and this clade is then divided into three main branches.
Let's start with the one that includes us, deuterostomia.
This clade includes hemichordates, echinoderms, like sea stars, and chordates. And it's important to note that Chordata is the only phylum in this entire category that includes vertebrates. Animals with a backbone.
Okay, then we have the two massive invertebrate clades. First, the Lophotrochozoa.
Right, and this name covers a hugely diverse group identified by shared developmental features. Some of them develop a feeding structure called the lophophore, which is a ciliated crown of tentacles. Others, like mollusks and annelids, pass through a unique stage called the trophophore larva.
And the third major branch is the ecdysozoa. This group is defined not by how it's built, but by a process.
Exactly. Ecdysis or molting. This clade includes nematodes and arthropods, which have to shed their stiff external skeleton or cuticle as they grow.
And now we should emphasize that this evolutionary tree is still, well, it's a dynamic area of science. There are ongoing debates.
For sure. The major one involves the position of the ctenophores, or comb jellies. For decades, sponges were the undisputed basal animal. But some molecular data suggest ctenophores might actually be the basal metazoans.
And that complicates things because leaf ctenophores have tree tissues and sponges don't.
It really does. If ctenophores are basal, it implies that tissues either evolved once and then were lost in sponges, or they evolved independently multiple times. That's a huge shift in how we understand early animal evolution.
And there's also the position of acola, these small, simple flatworms. If they are truly basal bilaterians, it forces us to reconsider the common ancestor of all bilaterians.
It suggests that ancestor may have been very, very simple, maybe with just a sac-like gut and an extremely simple nervous system.
Meaning the complex body plans we see today didn't just pop up all at once at the beginning of the bilaterian line.
Right, but rather they evolved after the split into protostomes and deuterostomes.
That's a powerful thought to leave you with. So to recap, the entire animal kingdom is defined by consumption, by movement enabled by specialized tissues, and a specific developmental blueprint.
Involving cleavage, blastula, and gastrulation. All this variation is contained within the architectural rules of symmetry, tissue layers, and body cavities.
And if you just consider the immense difference in complexity between, say, a simple choanoflagellate colony and a fully developed vertebrate.
The research tells us that this staggering disparity isn't necessarily due to evolving thousands of entirely new genes. Instead, it's about the regulatory power of ancient, tiny molecules like miRNAs and the subtle yet powerful rearrangement of ancestral genetic components.
Like adding that CCD domain to a cadherin protein.
Exactly. Evolution works by tinkering with the genetic tools inherited from the past, creating the stunning diversity we see today.
Thank you for joining us for this deep dive into the blueprint of the animal kingdom. We hope you feel thoroughly informed.